What is pangenome? And is it different from what we commonly know as the whole genome sequencing? Yes, it's different, and the difference is more important than it sounds. The whole genome sequencing gives us the complete DNA sequence of an individual genome. But one genome cannot capture every genomic sequence found across the species. A pangenome takes a different approach. It represents genomic sequences shared among individuals as well as sequences that vary between them. The central biological advantage of pangenomics is the reduction of reference genome bias. When sequencing reads are aligned to a single reference genome, divergent or absent regions may be missed. A pangenome provides alternative genomic paths, improving the discovery of novel genes and structural variants. For example, the Human Genome Project also used DNA from multiple individuals, but the resulting reference was assembled mainly as one linear reference sequence. In fact, the original reference was mosaic assembled from more than 20 individuals, but about 70% of the sequence contributed by one individual. The Human Pangenome Project takes a fundamentally different approach. Instead of collapsing genomic information into one main sequence, it builds high-quality genome assemblies from many genetically diverse individuals and represents their shared and alternative sequences together. The first major release included 47 phased diploid assemblies representing 94 haploids. So the key difference is not simply one individual versus many. It is one main reference representation versus multiple genomic paths represented together. That shift helps reduce the bias created when every genome is compared to just one reference. And this matters because genomic variation is much more than single nucleotide polymorphism. Pangenomes can reveal insertions, deletions, duplications, inversions, copy number variation, and presence-absent variations. Some of these differences can involve entire genes or large regulatory regions. And to understand this diversity, we can divide the pangenome into three broad components. First, the core genome contains sequences shared by most individuals. The second is accessory genome, contains sequences present in some individuals but absent in others. And finally, private sequences occur in only one or few individuals. And these variable regions can be biologically important, as they may contain genes of regulatory sequences involved in adaptation, disease resistance, stress tolerance, and other valuable traits. But representing all this diversity creates another challenge. A linear genome gives us essentially one path. A pangenome graph can represent multiple paths, allowing alternative sequences and structural variants to exist within the same genomic framework. And then pangenome-based CRISPR can take this diversity into account. Researchers can identify conserved sites, design allele-specific guides, and evaluate the target variation across different haplotypes. So, instead of designing for one genome, we can design with genomic diversity in mind, and this connects pangenomics directly to precision breeding. Pangenomics also can help identify favorable haplotypes from cultivated varieties or wild relatives. Those haplotypes can then be selected, introduced, combined, or potentially modified through genome editing. But finding a promising gene or structural variant is only the beginning. Its biological function still needs experimental validation. The pangenome tells us where to look. Functional biology tells us what actually matters. And the field is moving beyond DNA sequence alone. Further pangenomes will increasingly integrate genomic variations with transcriptomics, epigenomics, metabolomics, and phenotyping. Artificial intelligence may help also prioritize variants and predict their potential effects. So the question is no longer simply which genes are present. It becomes which genomic differences actually matters.